Thanks for the opportunity to have a quick chat about my research. I appreciate we're all probably quite fancying a coffee at the moment, so this is really a short, sweet one. Without further ado, it's just to give you an idea of some of the research that goes on in the centre, really. So, selection of oligonucleotide aptamers for Listeria monocytogenes. Okay, so first of all, what is an aptamer? Basically, an aptamer is a short-chained oligonucleotide. Okay, they have specificity for their given targets, and they are used in exactly the same way in molecular diagnostics as monoclonal and polyclonal antibodies. They aren't antibodies, in fact, they're quite different. We do tend to find that antibodies seem to perform better in terms of detection, just that slight bit better. That being the case, why do we bother with aptamers then? Well, there's a certain number of advantages that they have. First of all, the way they're produced. Aptamers can be produced in a test tube, whereas antibodies obviously require animals for production, or at least living tissue. By extension as well, that also reduces their costs, so aptamers are a little bit cheaper to produce. There's a fairly indefinite shelf life with aptamers also. A freeze-dried aptamer can last for donkey's years, whereas antibodies, being protein-based molecules, will degrade comparatively quickly over time. Another advantage, whether you're using an aptamer or an antibody, if when you get to the stage of using it in some kind of detection platform, very often you may wish to conjugate it with other molecules. What you tend to find is that when you label an antibody, you actually change the way the shape of the protein, and you will actually lose, the protein will actually lose target affinity, whereas that doesn't tend to be the case too much with aptamers, so they can actually be readily conjugated without too much loss to target affinity. The other advantage, of course, is also aptamers have a fairly limitless target potential, whereas antibodies, of course, you can only generate an antibody against something that elicits an immune response. So how does it work then? This is the method, systematic evolution of ligands by exponential enrichment, or SELEX. Just talk about this multicolored squiggly pool of worms here. Basically, that is a aptamer library. Within that aptamer library, what you have are oligonucleotide sequences. Every single sequence has fixed primer binding regions and a random region, potentially multiple thousands upon thousands of sequences, all with their own unique shape and, by extension, properties. Now, when you mix a pool of this variety with your targets, in this case cells, by sheer weight of numbers and purely essentially by chance, some of them will bind, a bit like in Kathy's talk earlier. They either like it or they don't. They either like the target or they don't. And from that point onwards, it's just a question then of removing the unbound sequences and retaining the bound ones, ready for further rounds of selection, in which perhaps the stringency of the binding conditions can be increased each round. So how do we do it then? We tend to use PureTac ready-to-go PCR beads. We add our aptamer library, we add our primers, we add some water, we PCR them up, and then we visualize them on gel electrophoresis. Now, with forward selection, so let's say you want an aptamer for Listeria monocytogenes, you can forward select and perhaps do maybe 8 or 9 rounds of selection, and then you've got your aptamer, so fantastic. But in actual fact, your aptamer may show quite strong cross-reactivity with closely related targets, such as E. coli or Staphylococcus aureus. So one potential way around this is a method called subtractive SELEX. So what you can actually do is, in the first instance, rather than mixing your aptamer pool with your intended ultimate target, mix them with a closely related target, and actually remove the sequences that do bind rather than retain the sequences that do bind. You then do that with Staphylococcus aureus and remove the sequences again. And then, of course, you can forward select then, and ultimately what you're yielding yourself is an aptamer that's specific for Listeria monocytogenes, but doesn't show cross-reactivity to closely related targets, like E. coli and Staph aureus. How do we actually separate them? Well, with targets as large as cells, it's reasonably simple. You just exploit the difference between the size of cells and oligonucleotides, or aptamers, if you like. So you allow them to mix in a bit of buffer, and then you can centrifuge them, and you create two distinct regions. You've got a region here with the cell pellet and the bound aptamers, and then you've got the supernatant, which is the unbound aptamers. So for forward selects, simply discard the supernatant, retain the pellet. For subtractive selects, the other way around, retain the supernatant, discard the pellet. In the aftermath of that, of course, you can in the aftermath of the selection process, you can obviously put them on the gel electrophoresis again, and you can physically cut the bands from the gel at whatever point you want, elute them from the gel, and then use them for further rounds of selection. So what sort of things are we interested in doing with these aptamers? Well, first of all, I mean, how far can Celex actually go? You may very well have a Listeria aptamer, but is it specific for Listeria monocytogenes? It's quite crucial in actual fact, because of the 10 known species of Listeria within the genus, only 2 of them are known to be pathogenic. So what use is an aptamer that can not distinguish between the pathogenic varieties and the non-pathogenic varieties? There's also the idea of live dead recognition also. If you can, pathogens in particular, the vast majority of them can only actually cause you any harm if they're alive. So it would be a useful tool to have aptamers that could recognize only live cells.